So today's video is going to focus a lot on evolution and how we get genetic variation and mutations, how those enable genetic variation. So we're going to talk first about some of our sources of variation. So mutations, the duplication of a gene, as well as chromosome fusion, those are all sources of genetic variation. Those actually provide the raw materials for evolution. They may not actually cause evolution to happen, but they're where we get that variation, which is what allows for the evolution to occur. So the mutations, gene duplication, chromosome fusion, these are our raw materials. This is what we're starting with. Okay? And from there, we've got meiosis and sexual reproduction. So meiosis and sexual reproduction, they're going to produce new recombinants of phenotypes. Basically, they're going to make new phenotypes. They're going to make things that have different physical characteristics. And with this different physical characteristics, now natural selection can occur. So the raw materials for meiosis to work with and the sexual reproduction to produce these recombinants came from mutations, gene duplications, chromosome fusion. But the actual evolution is going to occur with how those impact the phenotype. Once we mix all these genes together with meiosis, sexual reproduction, and we end up with new phenotypes, is that an advantageous trait that is going to carry on in the population? So mutations, as a general rule, many of them are not advantageous. They're deleterious or they are harmful. And a lot of mutations are also recessive in their mutation, okay, which is beneficial if it's a harmful mutation because it's harder to pass it down. Remember, mutations in somatic cells, so remember those are the regular body cells, these do not affect offspring. For a mutation to have an impact on the offspring, it must be in the gametes. Remember, the gametes are the sperm and the egg cells. If that mutation didn't occur in the sperm or the egg cell, it will not be passed on to the future generation. For instance, if I get skin cancer right now, that does not mean that my two daughters are going to get skin cancer. That was in a somatic cell. It was not in the egg cells that I passed on to them. Mutations can also either be at the gene level or the chromosome level. And so by that, you can have an impact on the actual gene, which is, remember, just those section of the DNA that codes for the one protein, or the entire chromosome, translocation, inversion, deletion. So we're going to spend the rest of our time on these different types of mutations. So our first kind of mutation that we're going to talk about is a point mutation. So in a point mutation, one nucleotide, remember that's our sugar, phosphate, nitrogen base. So 1A, 1T, 1C, 1G, okay? One nucleotide is substituted for another. So there's a couple of different kinds that we could have. We can have what's called a synonymous or a silent mutation. So these are the same. Okay, so your synonymous or silent mutations, those are the same. And so with these mutations, the substitution results in the same amino acid. So if this is my original over here with no mutation, even though I substituted one nucleotide, this G for an A, it still codes for lysine. Remember, we have that redundancy in the amino acid chart. When you're looking at that chart, and you'd have multiple things that coded for proline or multiple things that coded for alanine. Okay? So if you don't change the amino acid, then you will never know that the mutation is there. A non-synonymous mutation, okay, so our non-synonymous mutation can be a missense mutation. So with a non-synonymous mutation, this results in a totally different amino acid. And these are substitutions that usually occur at the first or second position in the codon. Remember, our nucleotides are grouped into threes, and that's how we read our codon chart. And so those first or second bases have a bigger impact than that third base. And so this kind of mutation is going to cause a change in the amino acid sequence. So in this case, the amino acid sequence is altered. And so that can do three things here. So we can get a protein that doesn't function as well as the original, that's going to be our most common. So I've changed the amino acids that are found in the protein. The protein doesn't work. The new protein doesn't work as well as the original one. That would be our most common kind of missense mutation. We could have a protein that functions better than the original. That is rare. That does not happen as much, to get a protein that works better than the original. Or we could have a protein that works just like the original. And that's usually going to be due to the R groups. If you think back to your organic compounds from your freshman year, and we'll review organic compounds later. But remember, amino acids have this, I'm going to come over here. Okay, so they have that NCC pattern, and they have an R group that comes off that middle carbon. That's what makes all the amino acids different. Okay, and so you can see, if you look at the amino acids down here, same, 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 same. It's what's coming off of that here that's different. That's the R group. And so if you're going to have a protein that's going to function very close to the original does, then that means those R groups are probably pretty similar. So with your non-synonymous or missense mutation, you get a different amino acid sequence, and so you could have one of those three scenarios. It's better, which is rare, it's worse, which is the most common, or it's exactly the same. You can also have what's called a nonsense mutation. And a nonsense mutation makes it code for a stop codon, which doesn't just alter the amino acid sequence by just one amino acid, it completely stops the formation of amino acids. If you had, for example, over here in the picture on our right, if you had a nonsense mutation here, there would be a stop codon and nothing after it. Okay? That actually stops synthesis of the protein. So that is a nonsense mutation. Point mutations in general are sometimes called substitution mutations. So that wouldn't matter whether it was a silent, nonsense, missense, synonymous, non-synonymous. It is still a substitution or a point mutation. So another kind of mutation that we can have is actual gene duplication. So in this case, the entire gene has been duplicated, and that gene occasionally can be moved to a whole another chromosome. So the gene has been copied and it gets moved to another chromosome. And so once that gene has been moved to a different chromosome, it can accumulate different mutations. And if the original gene and the copy of the gene are mutating differently, that can result in a completely different protein. So the example we have down here is the myoglobin protein. Okay, and so the myoglobin is the one there on the left. And so myoglobin binds with oxygen in muscles. Myo is muscle, and so it's similar to hemoglobin. Okay, so this binds with oxygen in the muscles. It actually has a higher affinity for oxygen than the hemoglobin does, which is why the hemoglobin will let go of the oxygen. And if you look at it, you can see some slight structural differences in it. In particular, right here, okay, they're a little bit different there. And so what has happened is myoglobin has been duplicated many times. And hemoglobin, over here on the right, is a result of a duplication of the myoglobin protein that has been the myoglobin gene, which has been mutated differently than the original one. And so that resulted in hemoglobin. Another kind of mutation we have is what's called a neutral mutation. So with a neutral mutation, you have a very slow, gradual accumulation of mutations that still allow the protein to fold into a stable structure. Remember, with proteins, it's all about their structure. When they lose their shape or their structure, they no longer work anymore. So even though these mutations are occurring, they are not altering the protein so much that they are now unstable. These are important for genetic change. They actually offer the basis of what we call the molecular clock, and you'll see that coming up on a future video. We talk about phylogenies and stuff, I think. We'll talk more about the molecular clock. It's basically just a basis to gauge the rate of evolution. So the example we're looking at here is what's called cytochrome, that's an R, C. Okay, so cytochrome C is a protein that we find on the mitochondrial membrane. Okay, so it's been around a long time. Remember, mitochondria are one of the original plastids. They have their own DNA. They've been around for so long. So the cytochrome C protein is coded for on the mitochondrial membrane. And it has the same function in both mammals and reptiles. But there are 15 amino acid differences between mammal cytochrome C and a reptile cytochrome C. And a different amino acid translates into a different mutation. So over time, these mutations have not affected the function of the protein. Remember, cytochrome C still has the same function, whether it's in mammals or reptiles. So it's gone through what we call neutral mutations. These mutations, even though they were occurring, still allowed the protein to fold into its stable shape and do its job. And so then you can use these mutations to gauge basically the rate of evolution. If you look at your diagram down here on the left, where you see where mammals and reptiles have diverged, okay, and mammals and reptiles diverged about 265 million years ago. And so if you calculate your 15 mutations had to have occurred between now and 265 million years ago, that's telling you that cytochrome C has mutated approximately every 17 million years. This is a slow process. Okay, what you can see up at the top here, this is our ancestral cytochrome C, again, sequence compared to the human sequence. So the ancestral one is the one here on the top, and the human one is the one here on the bottom. Okay, and so this comparison here shows that this gene is very highly conserved. This gene has not changed very much since its ancestral form, because those, what are called, remember those missense mutations? Okay, those occur more on what are called pseudogenes. And a pseudogene is a gene that has been duplicated, then mutated, and it's no longer functional. So that one can mutate away. It doesn't matter because it's no longer functional. This gene is no longer being transcribed or translated. That's why it's considered a pseudogene or fake gene. It is still part of the genome, okay, and it is still part of our DNA, but it is not transcribed or translated. Remember those introns and exons from your freshman year? If you don't, we'll be reviewing those as well. So still looking at these neutral mutations, let's look at a hemoglobin comparison. Okay, so there's an inverse relationship between the difference in amino acid sequence and how closely the organisms are related to humans. So basically, if I'm talking about an inverse relationship, the more differences I have, the further they are away from the their human relationship. So if we look at this comparison chart over here on this side here, okay, so we can see that gorillas have one difference, gibbons have two, and we work our way down, all the way down to a soybean, which has a similar protein in it called leg hemoglobin, because it's a kind of legume. Okay, and so this is showing us that out of the 146 amino acids that are on the human beta chain for hemoglobin, we have one difference between a gorilla. We are most closely related to a gorilla evolutionary-wise. 8 differences between a rhesus monkey. As we're working our way down this chart and we're increasing our number of differences, okay, we are getting further and further away from our relationships as well. Okay, and one of the things I want you to think about too is why in the world would a soybean plant have a hemoglobin protein? It doesn't have blood traveling through it that it needs to use to deliver oxygen everywhere. Okay, the benefit of this protein in these soybeans is so that it can remove oxygen from the plant so that they won't kill the bacteria. There are bacteria that live in the roots of the soybean plant which fix the nitrogen for it. Remember the nitrogen cycle? Those nitrogen-fixing bacteria that take the nitrogen that's in the atmosphere that the plants and the animals are unable to use and convert it into the nitrates that they can use. So the soybean is protecting those nitrogen-fixing bacteria so that they can provide nutrients for the plant. If we look at this diagram over here on the right, okay, it's showing the kind of the evolution of hemoglobin here. And so you can see there in humans there are 2 hemoglobin chains. There's the alpha hemoglobin chain and the beta hemoglobin chain. You see you've got alpha, beta. And so this is a result of that chromosomal duplication. And so these chromosomes have duplicated, and so they mutated differently. And so as you see these different mutations occurring here, it resulted in 2 different protein shapes, 2 different chains here. Okay, so again, though, showing that these mutations can happen and it is still able to do its job. It didn't lose its function. So our next kind of mutation is a frameshift mutation. You saw these also your freshman year. These are a result of an insertion or deletion of a nucleotide. They change the grouping of our codons. Remember, they change our groups of 3, whether we added something in and shifted it or we took something away and shifted it. But these change our groupings of our group of 3s, which means they can be changed the amino acid sequence from there forward. For example, if you're looking at this one down here, okay, so we've got our groups of 3s. And after the frameshift mutation, okay, you can see that now my groups of 3s are different. And so that's going to alter, as a general rule, that's going to alter my entire sequence of amino acids now. Most of these are also deleterious or harmful. Another kind of mutations are chromosomal mutations. So these are some form of complete chromosome rearrangement. These can result in a new species. They also offer another source of genetic variation. These are not just changing out an A for a T or adding one in there. We have totally done some gene rearrangement here. So we have a number of examples here. So the first one we have here is an insertion. And so in this case, we have added a section to the chromosome. So you can see here from chromosome 4, that section of chromosome 4 has been removed from chromosome 4. You'll see that's now shorter now, and added over here to chromosome 20. Okay, so chromosome 4 had some removed from it. Chromosome 20 had some inserted into it. Another example here would be a duplication. Okay, so we can see that over here. We've already talked about these. So we take a section of the chromosome and we duplicate it. And it doesn't necessarily have to go to another gene. It can stay in the same one. And now this chromosome is significantly longer because it has extra copies of the genes in it. Okay, we've got a few more. Some other kinds of chromosomal rearrangements would be an inversion. If y'all remember, again, you saw all of these things your freshman year. Remember, your inversion is when a piece of the chromosome basically breaks off, flips over, and gets reinserted right back in. You can see it very easily with the alphabetical way here. That DEF section was broken off, flipped over, and inserted right back into the chromosome, just upside down now. You can also have a translocation. Remember, a translocation is when a piece of one chromosome is translocated to another chromosome. Okay, so you can see here that here you can see easily with the color how they have switched. Okay, so they have taken non-homologous chromosomes here and switched portions of the non-homologous chromosome. And then this last one over here is an example of a fusion. This compares human and chimp chromosomes here. Okay, you can see that apes have 24 chromosomes, where humans we only have 23 pairs of chromosomes, and that's because you can see right here. Okay, the human chromosome number 2 is a fusion of chromosomes 2 and 3 on a chimp. So humans can have an impact on gene pools, just like we did in ecology. So what we're looking at, one of the main things we're looking at here is artificial selection. You can see that happening here in this picture with the mustard greens. Okay, so with our artificial selection, this is basically our selective breeding. Whether we're doing it with plants, whether we're doing it with the animals, whether we're looking for disease-resistant crops, the purebred horse, okay, and we have this artificial selection. You can see here all these different things we eat: cauliflower, broccoli, cabbage, kale, kohlrabi. Those are all versions of the wild mustard green. And what we've done basically is selected for certain characteristics. Kale it has enlarged leaves of the wild mustard green. So your cauliflower, the flowers themselves are sterile, and so that produces the cauliflower. And so the problems that can arise from this is we decrease the genetic diversity. And just like in ecology, when we decrease the diversity, we decrease the stability. For instance, a farmer could lose his entire crop. It's actually led to the formation of a seed vault that's in Norway where we store and maintain and preserve seeds to help preserve genetic diversity. We also see this with antibiotics and antibiotic resistance. Okay, we've got loads of organisms now that are resistant to most of our first response antibiotics. About 70% of pathogens out there do not respond to some antibiotic, not necessarily all antibiotics, but some of them. And so they become resistant due to human intervention. This basically overuse of antibiotics results in multi-drug resistant bacteria, or MDRs. And so the MDR bacteria, that's a T. So these multi-drug resistant bacteria are these superbugs. They don't respond to our first-line defense antibiotics. They're not responding to penicillin or amoxicillin. They're most commonly found in hospitals because that's where they have the most antibiotic exposure. And some of these organisms are going to be naturally resistant. Remember, if they're naturally resistant, then they're surviving, and so they're the ones that are reproducing. So that gene for resistance is staying in the population more, in the gene pool. And so they're difficult to heal from these. These organisms can attack internal organs, and so they are significantly more dangerous because we don't necessarily have any kind of drugs that can fight them off. Okay, so the side effects of reducing and eliminating the gene pools is these human impact things, they're going to encourage genetic drift. Remember, that's things like the bottleneck effect, the founder effect, things that basically decrease diversity within the gene pool. And we'll talk more with that vocabulary in other videos as well. This also diminishes gene flow, their ability to share genes. Okay, and so then we reduce genetic variation. And when we reduce genetic variation, that disrupts their ability to adapt. If all the phenotypes are the same, then it doesn't become better to have, if the environment changes, we may not be able to change with it because there may not be a different phenotype that is well suited for the new environment. The example over here are collared lizards. These are in the Missouri Ozarks, and these lizards, they're due to fire suppression because we as humans have been intervening and we don't want fires, and so that has reduced their habitat and has disrupted their gene flow, and so their populations are not mingling as well. So some of the effects of sexual reproduction can help combat some of this intervention and some of this lack of diversity. Remember, meiosis and sexual recombination are what encourage these new phenotypes, that natural selection can actually happen. So sexual reproduction is going to have these recombinants. It's going to recombine these genes in new and different ways. That's why offspring are all different, siblings are different, so that they have this variety within the population. It also helps result in unique offspring. So by having all these different offspring, we get the variety, we get the new combinations of alleles, and this will hopefully help encourage diversity within the population. So we're going to be working a lot with these different kind of mutations in class, okay, and applying them to the different phenotypes in evolution.